Welcome to class and test Superfast division class Eukaryotic cells. Eukaryotic cells vary greatly in their size, which may be in the range of 10 micrometer to 500 micrometer. यूकैरियोटिक सेल्स में हम लोग स्टडी करेंगे वन पार्ट है प्रोटोप्लाज्मा मेम्ब्रेन, ऑल्सो सेल वॉल इन प्लांट सेल्स, और दूसरा पार्ट होगा हमारा प्रोटोप्लाज्म। प्रोटोप्लाज्म को दो पार्ट होते हैं, न्यूक्लियस और साइटोप्लाज्म, और साइटोप्लाज्म में दो पार्ट होते हैं, हाइलोप्लाज्म और साइटोप्लाज्मिक स्ट्रक्चर। साइटोप्लाज्मिक स्ट्रक्चर्स में टू पार्ट्स हैं, एक है सेल ऑर्गेनल्स, दूसरा है सेल इंक्लूजन। सेल ऑर्गेनल्स में आ जाते हैं एंडोप्लाज्मिक रेटिकुलम, गोल्गी एपरेटस, राइबोसोम्स, माइटोकॉन्ड्रिया, प्लास्टिड्स, लाइसोसोम, सेंट्रियोल्स, बेसल बॉडी, माइक्रोट्यूब्यूल्स, माइक्रोफिलामेंट, सीलिया एंड फ्लैजला। और सेल इंक्लूजन्स में आते हैं हमारे को वैक्यूल्स, रिजर्व फूड, गैसेज, ईटीसी। ओके, every living cell is externally covered by a thin transparent electron microscopic elastic regenerative and semi permeable membrane called cell membrane. Another name of cell membrane is plasma membrane या plasma lemma, present in both prokaryotic and eukaryotic cells. The plasma membrane and all the membranes covering various cell organelles present inside the cell have same intrastructure and are collectively called biomembranes because they play vital functions for a living cell. It regulates the exchange of materials between the cytoplasm and extracellular environment. It also helps in synthesis of many macromolecules response of the cells to a variety of physical and chemical stimuli etc. And biochemical composition of cell membrane. Best material for the study of biochemical composition is human RBCS. Plasma membrane is formed of lipids, about 40%, proteins, about 58 to 59%, and carbohydrate, about 1 to 5%. Lipids. Lipids are mainly formed of phospholipids and cholesterols. Phospholipids constitutes about 55 to 75% of the lipids. Cholesterol molecules form about 25 to 32% of total lipids and lie between the phospholipid molecules. Lipid molecules are arranged in a bilayer in the cell membrane on the basis of amphipathic nature of lipid molecules. Amphipathic nature. Polar or hydrophilic head of or glycerol of the phospholipid molecules of two layers are directed in opposite directions and interact with aqueous environment on both sides. Non-polar or hydrophobic tails of two layers face each other. This arrangement forms a water-resistant barrier. Lipids. More common types of phospholipids are lecithin, most abundant cephalene, phosphatidyl serine, phosphatidyl ionostol, and phosphatidic acid. Phospholipid. Phospholipid molecules form the structural framework of plasma membrane and allow the faster movement of fat-soluble molecules. Cholesterol molecules stabilize the plasma membrane. Proteins. These constitute about 50 to 59% of plasma membrane. Singer 1971 demonstrated that these membrane bound proteins are of two types. Number one is peripheral or extrinsic proteins, and number two is intrinsic or integral proteins. Number one, peripheral or extrinsic proteins. These lie outside the outer and inner surface of phospholipid bilayer and are loosely attached by weak electrostatic bonds. These form about 30% of the proteins, peripheral or extrinsic proteins. Intrinsic or integral proteins. Now let's study intrinsic proteins या integral proteins. These are partially or completely embedded in the phospholipid bilayer. These are not easily separable. This form about 70% of the protein. 70% of the protein is intrinsic protein या integral protein. Structural proteins form the backbone of membrane and provide elasticity and mechanical stability. Enzymatic proteins, that is magnesium plus plus activated ATPase, sodium plus potassium plus activated ATPase, alkaline phosphatase, RNase etc. regulates cellular activities. These are enzymatic proteins. Carrier proteins help in facilitated and active transport. Hormone receptor proteins help in the action of quick acting hormones, that is epinephrine. Third is the carbohydrate, about 1 to 5% of chemical composition. Main carbohydrates are hexoses, hexosamine, fucose, celiac acid, etc. These are present as glycoproteins and glycolipids. Structural models of plasma membrane. Number 1, sandwich model proposed by J. Danelli and H. Davison in 1935. Unit membrane model proposed by J. David Robertson, and fluid mosaic model proposed by S. Johnson Singer and Garth Nicholson in 1972. Most accepted model, sandwich model. Hydrophilic zone, and in the between the hydrophobic zone. Sandwich model. Plasma membrane is a lipoproteinous and trilaminar or tripartite membrane. It is formed of outer and inner denser protein monolayer and a middle transparent phospholipid bilayer. So the plasma membrane is a protein lipid protein sandwich in the sandwich model. B. Protein molecules are globular alpha proteins and give stability to the plasma membrane. Heads glycerol of phospholipid molecules are directed in opposite directions and are associated with protein molecules, while the tails fatty acids of two layers face each other. Plasma membrane is a porous membrane. It has a microscopic pores of 7 to 10 angstrom according to sandwich model. Drawbacks of sandwich model. It doesn't explain the functional specificity and variability in the biomembranes, and it does not explain active transport. Unit membrane model. The next model is unit membrane model, lipid bilayer and glycoprotein coat. Unit membrane model. A. Biomembrane is a lipoproteinous and trilaminar. Protein molecules are extended beta proteins or fibrous and not globular. Each phospholipid is a polar molecule with a hydrophilic head of glycerol and a hydrophobic tail of two fatty acid chains. Heads of phospholipid molecules are directed in opposite directions, while tails of two layers face each other. Average thickness of biomembrane is 75 to 100 angstrom, in which each protein layer is 20 to 30 angstrom, while phospholipid bilayer is 35 to 40 angstrom. Lamellar models. Both Danelli and Davison model in 1935 and Robertson's model of unit membrane are collectively called lamellar model. Fluid mosaic model proposed by S. Johnson Singer and Garth Nicholson. It states that plasma membrane is formed of a protein icebergs in a sea of lipids. Plasma membrane is lipoproteinous and trilaminar. Phospholipid bilayer is fluid in nature, in which phospholipid molecules show two types of movements. Number 1, transition movements. Molecules change their position with the same layer. Number 2, flip-flop movements. Molecules of two layers can be interchanged. This is the structure of plasma membrane. Proteins are globular and are of two types. Number 1, extrinsic or peripheral proteins, and number 2, intrinsic or integral proteins. Some intrinsic proteins are so large sized that these project on both the surfaces of phospholipid bilayer and are called tunnel or transmembrane proteins. These act as charge channels for the passage of water soluble materials. Each phospholipid molecule is a polar molecule and has two specific ends, hydrophilic head of glycerol and a hydrophobic tail of two fatty acid chains. So it is an amphipathic molecule. Tails protect from water as are hydrophobic. Heads of phospholipid molecules of two layers are directed in opposite directions while tails of two layers face each other. This arrangement forms a water-resistant barriers. Carbohydrates, especially oligosaccharides, are associated with only outer surface of plasma membrane. These either form from glycolipids or glycoproteins. Cholesterol molecules are present in between the phospholipids. These provide stability to animal membranes, and due to this quasi-fluid state, biomembranes can undergo dynamic changes and help in cell growth, formation of intercellular junctions, secretion, endocytosis, cell division, etc. Plasma membrane of a bacterial cell. Now let's study the plasma membrane of bacterial cells in the next class. Thank you.